Yes. Hello, everyone, and welcome to this year's Copenhagen Life Science Summit. We are glad that so many wanted to participate, even though we had to go digital this year. We are really glad, however, that the station on Frederiksberg wanted to host us. So yeah, we have put together a really interesting program around the whole COVID situation, where we are focusing on the fast tracking vaccine development, and together with Bavarian Nordic and University of Copenhagen and Danish Medicine Agency, we put together a really interesting program. My name is Andreas, and this is Maya, and we will together today be your host for this program. And we are so happy that to see all of you participating. And now I want to give the word to Maya, who will introduce this year's program.
Yes. Yes, so as Andrea said, we have designed a very thorough program for you to be able to cover all aspects of vaccine development. First off, I will give a short introduction to Synapse and what we are and what we're doing. And then I will give the word to Morten Agertog Nielsen, who is an associate professor at the Department of Immunology and Microbiology at the University of Copenhagen and also co-founder of AdaptVac, who is currently working on the Danish contribution to the COVID-19 vaccine program. And after a short break, we will have Tommy Kaino, who is the Executive Vice President and CBO at Bavarian Nordic. And he will take you through the industrial contribution and aspects of vaccine development. And lastly, after a short break, we will have Thomas Senderovich, who is the Director of the Danish Medicines Agency, and he will cover some of the aspects of governmental regulatory processes that will secure the optimal safety of the final product before it's introduced to the market. In the end, we will have a panel discussion where we can discuss some of the topics that the speakers have covered in their presentations or anything else that you might want them to discuss. So we strongly encourage you to all send in your suggestions to anything you want discussed in further detail. And you can do that via Slido. But off to Synapse. So And Synapse was founded based on the fact that 93% of life science students do not think that companies provide sufficient information about what competencies are needed in the industry and what they can use their academic education for. So that led to the formation of Synapse in 2014. We are a student-driven non-profit organization in the Nordics that has the goal to bridge the gap between academia and industry in life science. And we do that by creating events, workshops, and networking opportunities for students and recent graduates with an interest in pursuing careers in the life science environment. We inspire, develop, and connect to fulfill our vision. We inspire our participants through knowledge lectures, career fairs, and meet and greet events, such as the Copenhagen Life Science Summit, which we are hosting today. We also develop our participants through workshops, competitions, and mentoring opportunities. This could, for example, be our Biobusiness Summer School. Then we connect people by providing this platform where people can meet over mutual interests, so we can connect the future of life science. Synapse has recently also launched a hub in Lund, so marking our international presence. And we can continue to bridge the gap between academia and the industry, also across Øresund. So we are very happy to also be present in the Medicon Valley cluster. So now Synapse has 3 hubs in total, located in Copenhagen, Aalborg, and Lund. We arrange over 30 yearly events with over 2,500 participants. So really we reach a large number of life science people in the life science community. Our 5 flagship events include the Biobusiness Summer School, which has 60 participants from numerous countries. It takes place over 5 days in August, and we invite over 20 speakers with a broad variety of life science backgrounds, and we also strengthen our participants' practical skills by hosting a case competition in collaboration with BCG. Then we have the Copenhagen Life Science Summit, which we are presenting today a short section of what the usual physical version of the Life Science Summit is. And for that, we usually have 300 participants over a day. And then we have the Connect Medicon Valley, which is a 3-day showcase tour of the upcoming amazing science in the Medicon Valley cluster. And then we have the Science Mentorship Program, which has 30 mentees who are paired with a mentor, and that spans over the academic year. And lastly, we have the Life Science Career Fair with over 500 participants, where people can meet potential new employers and hear about their what they seek in their employees. But really, Synapse is much more than that. We have over 30 events annually, and you can take place, or you can participate in them if you follow us on Facebook, LinkedIn, and Instagram, where we have connected the largest network of life science students in the Nordics. Of course, all of this we would not be able to do without our partners, which we are very grateful to have and will contribute to us. And of course, the Synapse team, we're all very ambitious and dedicated, and we work long hours free of charge, but it's really all worth it because of the great team spirit we have. To highlight one of our latest accomplishments, we hosted an online career fair in September, which was really a great success. We had over 500 participants and 17 companies, and many conversations and job applications were sent at this career fair. It really worked very well, even in an online format. Also recently, Synapse moved to new headquarters here at Station, where we are now, and it has really, it's a really great for Synapse to develop us as a student organization, being in an ecosystem with similar student-led organizations, to keep getting new insights and feedback to how we run our large-scale organization. Synapse has been recognized for our efforts by receiving several awards, and we have also been featured in Nature Biotechnology and the Medicon Valley Yearbook, as well as received grants from larger funding bodies in DK. Great. But that was all of it from me from now. If you have any queries about Synapse and what we do, you can always write to our email or follow us on Facebook, LinkedIn, and Instagram. And you're also welcome to check out our website to read more about any of our events. But now we should get to the real topic of today, so I will give the word to Morten Nielsen.
Thank you for inviting me. I don't think I need this one.
No.
So I am, can I get the clicker? And the presentation is this one. I deliberately put 2 title slides because I'm going to talk about taking a vaccine from the lab to the real world. And our technology was invented at the university and then later spun out in a small company where we can then take the vaccine and make a contribution to health sciences and health in community. Our technology is in basic quite simple. It consists of producing a capsid particle that resembles a virus. On this capsid particle, we can attach antigens from different pathogens. We can express these antigens in different organisms, and that makes a very unique and versatile platform. The attachments of antigen is based on a covalent bond. Actually, it's a protein from a bacteria that has been split into two domains, a tag and a catcher. And when these two domains come in close proximity, a covalent bond is made between the amino acids in these two domains. So we can fuse either the tag or the catcher into the capsid or antigen. And then when we mix the capsid with the antigens will assembly onto the surface of this capsid particle. In this way, we can make non-dangerous antigens in solution to take the form of a dangerous virus-like particle, shown here in this cartoon-like picture. Actually, we have an example, a very good example of a capsid-like particle vaccine, and that's the HPV vaccine that protects against cervical cancer. That particle is, as ours, produced by expressing a small subunit that then self-assemblies into this capsid particle. In the beginning of the clinical development of HPV, 3 doses were advised. Now, actually, the WHO are looking into only 1 dose, and that is because the antibody response towards this capsid particle of HPV is very, very long-lived. We have antibodies after 1 dose regimen up to 8 to 10 years in the plasma of immunized individuals. And that's what we are trying to mimic with our capsid-like particle. The immunology behind this is quite complex, and I'll not go into details with this slide here. Just to say that there are different kind of virus-like particles, the capsids and then the lipids. In the lipids, the enveloped virus-like particles have the antigens floating free in the lipid membrane of the envelope. And that generates a different kind of antibody response. That is, for example, the AstraZeneca Oxford virus-like particle. That's an envelope virus. In our case, we have a very strong and tight capsid particle, and that generates another antibody response. So some soluble proteins are very low immunogenic in nature, and it's very difficult to generate an immune response against them. Other soluble proteins are very immunogenic, and when we take these very low immunogenic antigens and put them on the surface of our capsid, in all of these 4 examples I have here, we have generated a very, very strong antibody response that we could not generate with the soluble antigen and an adjuvant alone. Actually, the response we get to the antigens attached to the particles is so strong that we can generate antibodies against self-antigens. Self-antigens are antigens that you normally have in your body, like albumin, for example. In this case, we have used the HER2 antigen, which is associated with cancer, expressed a lot in breast cancer. And if you generate an antibody response against HER2, you can clear the cancer. That is shown here in this example from GSK, where they developed a vaccine based on HER2, and they had a very low level of responders. But in the case where they had responders, the cancer was cleared. And we think we can mimic this vaccine, but just in a better way, generating a very strong antibody response against the HER2 and clear cancer. Actually, we think that we might substitute a lot of the monoclonal antibody therapies that are currently in the world, and which is a very growing field. And we can substitute this by a low dose of vaccine, so that the body becomes the antibody factory and can help you clear the disease. This is especially important in cancer and chronic diseases. So just to summarize, what we think we have with our technology is a very rigid structure. We have a dense display of antigens, and we can display very large antigens on the surface of the particle. Also, they are unidirectionally presented. That means we can focus the antibody response against the important epitopes of the antigens. And that, in contrast to some of the other virus-like particle technologies, generates a very long-lived antibody response. And that is what we are going to test now with our COVID-19 vaccine. And this has really been a fast track for us because we started in April, and now we are we are actually filling the vaccine into glass vials next week. Again, we are taking part of the pathogen and putting it on our particles by this covalent coupling. We are working with the RBD domain of the spike protein of the SARS-CoV-2 antigen spike protein. And that is the green part here. And that is the part of the spike protein that attaches to the human cells and allows for virus entry into the cells. If you target this domain by an antibody, then the virus cannot penetrate the cells. So we express the RBD domain in Drosophila S2 cells together with expression biotechnologies. And we verify that we have the correct fold of this antigen by doing affinity measurements so that we can show that the antigen binds to the S2 receptor. Thereafter, we test the immunogenicity in mice, and we show actually that the RBD antigen is one of these very low immunogenic proteins. When you mix it with a strong adjuvant, you barely get an antibody response against it. But shown in the orange, we do get high levels of antibodies when we attach it to the particle. The last form of functional assays we do is the virus neutralization assays. We show that we get a very, very high induction of neutralizing antibodies against SARS-CoV-2. Actually, attaching the antigen to the particle augments the response, the virus neutralization response, by 500 times compared to the soluble antigen. And we get virus neutralization titers in the order of 1 to 60,000 times. That means we have to dilute the serum from the mice that we're testing 60,000 times before we see 50% entry of virus into the cells. That is a very high dilution of serum. And actually, when we compare this to other vaccines, shown here are the RNA and mRNA vaccines in the middle, the green and red box. And that is the Moderna and the BioNTech vaccines. That's their preclinical data. And they show 90 to 95% efficacy, at least shortly after the immunizations. When we compare to that, our virus neutralization is an order of magnitude higher. Actually, we think that we might be able to sustain and support the WHO target product profile of having a one-shot vaccine that can be distributed across the globe, because our vaccine appears to be stable in high temperatures, oppositely of the mRNA vaccines. Shown here to the right is a one-shot vaccine in mice and the virus neutralization titers in orange. And compared to the blue, which is antibody titers from SARS-CoV-2 severe patients that have recovered, we show that we can generate with 5 micrograms in a mouse the same level of antibodies as in patients that have had the disease for several weeks. We have been focusing on upscaling this and take it from the laboratory into a factory, together with AGC Biologics. We know that we have the capacity to express the particle in very high amounts, and also the antigen. We need to demonstrate that we can also purify these high levels of expression before we can go further in the clinical development. But based on the capacity right now, we could produce several billion doses each year of this vaccine. So these are the milestones from April to now. We have made a license with Bavarian Nordic on the further clinical development of this vaccine, and they have supported us to a very great extent through this last half year, having all the regulatory consultancies in-house. So with this, I want to thank you for inviting me here. It's a great pleasure being here, and I really appreciate initiatives like this. Thank you.
Really a great tour through the molecular tools you can use to make a vaccine. We have not received any questions so far. So I think we will just have a short break.
Yeah.
And then you can go out and get a cup of coffee or get some fresh air. There's a question. Okay. So it says, I suppose the patient response data is from clinical trial. Are you able to comment on the sample population for the data?
It's a bit difficult to see. We haven't started our clinical trial yet. It will start in Q1 2021. But the response that we're comparing to in the human data, that's from Danish patients. Yes.
Great.
So the question is how the antigen is coupled to the VLP, and that is by this catch-attack interaction. It's a split protein that actually the bacteria is living in a very harsh environment, normally in the gut, for example, and they have these interdomain isopeptide bonds that make the proteins very stable. When you engineer these protein, you can separate them in two, and they have very high affinity to each other, and then when they come in close proximity, they make this isopeptide bond between two amino acids, and that is basically unbreakable. We expect our vaccine to be very safe. The other vaccines based on recombinant protein expressions are in generally very safe. Yeah, so the question is how long the virus neutralization antibodies are in the mice, and we do not have data for that yet. And actually, the mouse is not a very good model to investigate longevity because they don't live for very long. A better model for that is non-human primates that live for a longer time and have a more similar antibody response as humans. So the question is regarding advantage compared to mRNA. Maybe I can comment on the disadvantage first, because our technology is somewhat slower. We need to produce the protein outside in a fermenter and purify them, and you need to verify that you don't have any unwanted items in the mix. With the mRNA vaccine, you inject the RNA and the body starts to produce the protein. So that's a very, very fast technique. What I have looked into the literature for mRNA vaccine, and it appears that the antibody response is waning faster with these mRNA technologies compared to ours.
Okay. No further questions? Okay.
So I would say the question is regarding how many proteins on the capsid is needed, and actually you cannot say a number because you need to have the antigen packing the particle, and the larger the antigen you have, the less packing you can have of the antigen. The particle consists of 180 subunits, and for small antigens we have 100% coupling. For this vaccine we have around 40% to 50% coupling, and that is because the RBD domain of the SARS spike protein is a big antigen.
That was all the questions for now. If any other will pop in, we will take them later during the panel discussion.
Okay.
But thank you so much
Thank
for
you.
now, Morten. Morten will also be here later if you have any other questions. But yeah, as mentioned, we'll have a short break, and you can go get a coffee or some fresh air or whatever you'd like, and we'll be back in a few minutes.
Hello, everyone, and welcome to the second session now with Tommy Kaino from Bavarian Nordic. Before I will introduce him, I just want to encourage you all to send the questions throughout his speech, because Slido has this amazing feature where we can upvote and downvote good questions. So Tommy would get the best question after his speech. But yeah, Tommy is Executive Vice President and Chief Business Officer at Bavarian Nordic, and we'll talk a bit about novel strategies to speed up the clinical development process. And I'll let the table up to you.
Yeah, thank you. And it's great to be here. I've had the privilege of being in one of these life science events as a speaker before. And that was when there were 300 people in the audience. It was an amazing experience. But I'm sure even with this virtual kind, the organizers have made a great effort to make that experience similar and as exciting. So again, very happy to be here. And I'll be talking about accelerating vaccine development. And I think one of the important things to remember and remind ourselves that vaccines are among the most important tools in public health. And if you all remember a US president called Franklin Roosevelt, he's the only president in the US who served for 3 terms, was actually elected for the 4th term. He came from a very wealthy family. His cousin was one of the former US presidents at the time. He lived in the 40s, but he got polio and got paralyzed and spent the rest of his life in a wheelchair. So, and why I'm bringing that up is that polio was still quite common, even in rich countries like the US, even among very wealthy people in the last century. And now there's 0 cases of polio in the US and in many other countries, just because we have a very effective vaccine there. Smallpox, one of the diseases that probably has had the most impact on the development of civilizations, in fact, wiping out most of the indigenous populations in the Americas, and having an impact also in other parts of the world. Again, it's wiped out as a disease. There is no smallpox anymore in the human population. The only example or samples of the smallpox virus are in freezers in the US and Russia. And again, because of a very effective vaccine. And then the other thing that we need to remind ourselves that effective vaccines are not enough. People need to trust these vaccines and actually get vaccinated. And there, for example, measles, where there has been a vaccine, a very good vaccine for years, it has brought down the number of cases, and this is an example in the US of measles, but we still have outbreaks, both in the US, but also in various European countries. And those outbreaks occur because people, for whatever reasons, do not get vaccinated themselves or don't bring their children to be vaccinated. So it's for all of this to work and to really eradicate diseases such as the ones we see on this slide, but also other diseases like the pandemic that we're experiencing now. It comes to 2 things. We need effective vaccines, but we need people to trust the vaccines and actually get vaccinated. And that's why vaccine development is highly regulated. In the case of the smallpox vaccine, the original one, which was already developed at the end of the 17th century, that clinical trial, if one can call it as such, the first thing consisted of Edward Jenner scraping off some pus from a cow's hide. That cow had been infected with cowpox, and taking that pus and putting it on the arm of a small dairymaid or a child, and actually then challenging that with sort of smallpox and finding out that that person was immune against smallpox. Now, that type of clinical trial, I'm pretty sure, would not occur these days. It's very regulated and starts from the non-clinical research that's done in animals, small animals, mice, as Morten was just talking about, non-human primates, and toxicology studies in rabbits before the regulators give the authority to companies and academics to take those vaccines into clinical trials. And again, it's highly regulated, phase 1, phase 2, phase 3, and I'm sure Thomas in the next presentation will talk more about the process. And now, sometimes people think that companies, pharmaceutical companies, companies that produce vaccines don't like this regulation, that somehow they'd want to avoid it. But that's not at all the case. Actually, the regulation is very good, because if it wasn't there, there would be more examples, as is the case for RSV. So back in the 60s, there was a vaccine developed against RSV, which is the respiratory syncytial virus, which is very common in newborns. So they get very sick because their alveoli in the lungs get constricted and they're put in emergency rooms, and it's a very difficult disease for newborns. There was a vaccine developed. It was an inactivated virus, the RSV virus. It was inactivated using formalin, and it seemed to work really well. It sort of passed the first safety measures. But then what turned out is that actually, rather than protecting the children against the disease, the children who got the vaccine got even a worse case of RSV than the ones who didn't. It's a phenomenon called enhanced disease. And that basically put a stop to RSV vaccine development for some time. And now it's only very recently that companies have started again to look at different vaccine candidates, such as our company looking at our vaccine against RSV, and utilizing different technologies to overcome both safety and efficacy challenges. But that's really why effective regulation and the rigorous process in vaccine development is so important, because if these types of failures, for example, for RSV and in the 60s, can cause significant delays and impact the whole development process also of other vaccines. Now, that was safety. The rigorous process is also very important when it comes to the efficacy of vaccines. And there is a very significant difference between having a 99 or even % effective vaccine, as is the case for some diseases, like measles here, or 40 or 50%, as is the case for example, a flu vaccine. And in this slide, what's shown here is the different diseases, how they're transmitted, and then the R0. And that R0 means that if one person who has the disease, how many people on average does that person infect? So the higher the number, the more potential the disease has of spreading in the community. And you'll see measles is a very high R0, so every person who has measles on average spreads it to 12 to 18 people. So you need a highly effective vaccine to stop the flow of the disease. And of course, the less people one infects on average, so for influenza, 1.5 to 1.8, you can get by with a less efficacious vaccine. If one looks at COVID-19, which is on all of our minds these days, we don't really know what the R0 is in the natural environment. We now have all of these social restrictions that have brought that R0 down, but if it was left to spread on its own, how high would that be? So we actually don't really have a good idea how effective a vaccine we would need to control the spread. Vaccine development takes time. It took 105 years after we discovered what causes typhoid to develop a vaccine against it. Polio, 47 years, and so on. And it's only in the recent years that we've gotten these timelines compressed. And if there is a sort of a silver lining to this cloud that COVID-19 has brought on the world, it is that we've surfaced a number of technologies, new platforms that have proven effective to be able to generate new vaccines, efficacious vaccines, and just to pick out the mRNA vaccines that have shown great results from Pfizer, BioNTech, and Moderna. Using a technology that was only really developed in the last decade. So before mRNA, if it was injected by itself, really caused a very bad sort of immune response on the skin, and you couldn't use it. But through work done by various scientists, including Katarina Kaslowska, in the mid-2000s, they identified a way to stabilize the mRNA molecule in a way that you could sort of bring it into the body without those type of side effects. And then there's been a lot of development how to encapsulate the mRNA so it doesn't degrade as it's put in the body. And now we're seeing that it actually can produce these vaccine factories. You basically use the human body as a factory to make the vaccines, which is really amazing. So we've just seen a lot of scientific progress in just the matter of a few months during 2020. But there are other technologies in addition to mRNA. We've seen recombinant proteins, as Morten was describing. We've also finally started to see viral vectors being utilized effectively in vaccine development. Janssen and ourselves, Bavarian Nordic, we had recently this summer the first viral vector vaccine using adenoviruses approved against Ebola. And now, as many of you know, adenoviral vectors are being used to develop SARS-CoV-2 vaccines, also AstraZeneca among one of them. And we are seeing the results of all of this technological and scientific progress, where it took 20 months in 2003 when SARS won. We had identified the genetic sequence of that to when the first human study of a vaccine took place. Now it was a matter of 45 days. Now, obviously, it helped that we knew that it was spike protein from the first SARS that would prove effective or could prove effective against SARS-CoV-2. But nevertheless, that technological progress in being able to identify the optimal antigen, having the sequencing capabilities, being able to fast translate that sequence into a viral protein, either using mRNA technology or a viral vector, showed that these technology platforms, vaccine platforms, can be applied also against other future emerging diseases. CEPI, which is an international organization that was created to combat emerging infectious diseases, calls these disease X. So we don't know what it will be this year or last year. It was COVID-19. Whatever that will be in 5 years or 10 years, we don't know. But with these types of vaccine platforms, we should be able to quickly develop novel vaccines to combat those diseases, hopefully in even a shorter timeline that this record-breaking vaccine development during this year has been. But technology is not enough. It's also the support from the regulatory agencies. And again, Thomas will talk, I'm sure, a lot more about this. But what the EU, as an example, has done in their COVID-19 response, they set out with regulatory enablers, enabling companies to progress their clinical development of their vaccines in a safe, rigorous way by cutting through bureaucracy and enabling scientific advice. So early engagement with the European Medical Agency and international cooperation, so that scientific knowledge was shared and companies benefited from it. An accelerated procedure for authorization, again rigorous, not cutting any corners, but making sure that things move fast. And then some flexibility in relation to labeling and packaging requirements, so not needing to have the package with the language of all 26 or 28 European countries, but sort of making some flexibility or having some flexibility there. And then importantly on the legislation around genetically modified organisms. So there's been a lot of government support and European support for enabling this fast progress, and that needs to be recognized. But as said, it's not just the efficacious vaccine. People actually need to get vaccinated. And that starts with people being willing to vaccinate it that I talked about previously. They need to trust the safety of the vaccine. They need to trust the efficacy of the vaccine. So they need to get in line to get vaccinated. But if the vaccine doesn't make it from the manufacturer, the manufacturing plant, to whatever site, whether it's a tent, or whether it's a school, or whether it's a doctor's office, and be there so people can get vaccinated, this will not happen. Previously in my career, I was a doctor in the Finnish military, through military service, and I had the opportunity to sort of vaccinate the new recruits as they walked in. So there would be 1,000 new recruits coming in, and we stabbed them from all sides with different vaccines. And with just that experience, you know that these mass vaccination campaigns will be quite challenging to get done on time, and making sure that you have the exact amount of vaccine that you need there, it's distributed optimally, and it's stored optimally, both in the storage spaces and then during the supply chain, but then also in the actual vaccination site. And if the vaccine requires minus 70 degrees Celsius of storage, that's a difficult thing. There are not so many minus 70 freezers out there. Hospitals have it, doctors' offices typically don't. If it's minus 20, that starts to be a little bit easier. But again, if we're talking about not countries like Denmark, but India, Africa, then that cold chain becomes a problem. And it's not just the distribution itself, it's the manufacturing. mRNA, it's never been manufactured on a scale that's needed now, fortunately enough. The technology is quite simple to manufacture, so that should be good. But then for viral vectors, again, that demands certain type of technology and manufacturing processes. So there's a lot of effort being put by companies to ramp up to a scale that's needed. And that's really one of the reasons that I just mentioned why our company, Bavarian Nordic, started the cooperations and took on the license from AdaptVac on their SARS-CoV-2 vaccine. And Morten there sort of talked a lot about the preclinical data and the animal experiments around the vaccine, but just to sort of highlight some of the key reasons why we think that this platform is such an interesting one and why we chose to take the license on it. As said, it's a flexible one. So you can sort of imagine if the virus starts mutating and we want to adapt the vaccine because we need to change the antigen, or whether there's that future disease X that we need to design a new vaccine against. It can be done quite quickly and effectively, and utilizing the same type of manufacturing technology going forward. The other benefit is that it can be manufactured, we believe, and the early data supports it, in a very high scale. So both the VLP itself produced in E. coli, which is a simple process, as well as the antigen and S2 cells. And utilizing high-capacity bioprocessors, you should be able to get doses out on a very high scale. And it's the storability to make it and stability to enable a smooth supply chain seems to be quite good as well. So let's see when the sort of data starts accumulating and as time goes by, but I can be quite confident saying it's not going to be -70, it's not going to be -20 that this vaccine needs, but it's going to be much closer to temperatures that we are more economical and comfortable with in the supply chain. But most importantly, these things combined make it that it will have the target product profile of the WHO recommendations. So it should be highly effective. We really hope, and the early data supports this hope, that it would be a single dose regimen. So not having those millions, hundreds of millions of people going to get vaccinated, not once, but twice with some of the other vaccine technologies. So having just a single dose. We expect to be rapidly protective. Storage conditions we talked about. And again, if we're talking about vaccinating many people, that low cost of goods through high scale is very important. But most importantly, we believe it will be a very highly efficacious vaccine. And obviously that needs to be proven in clinical trials, which hopefully will start soon. But the animal data that we have, both from mice and this data from non-human primates, shows that with either with a higher or lower dose of the vaccine and vaccinating non-human primates, extracting sera from them and looking at neutralization antibodies and neutralization activity, we see even with one shot of the vaccine, it correlates with convalescent sera extracted from SARS-CoV-2 patients. So that's why we continue to believe this is a very good vaccine. We continue to see a real role for this vaccine in fighting COVID, also in the long term. So with that, I thank you. And if there are any questions, I'm happy to take them now.
Thank you for a great speech. I can tell you we have a lot of questions.
Okay.
So you made an impression. So the first one is from Julia. How do you evaluate the severity and prevalence of COVID-19? And do you think that other diseases with similar prevalences would get this priority?
It's a great question. I think and I'll share my own thoughts on it. Back in February, March, I would have said that we're looking at a sort of a flu-like thing, which doesn't seem to be quite as dangerous. Now, this thing seems to spread. I mean, we've seen that even in conditions where a lot of society is barely functioning because of social restrictions, many of us have not traveled outside the country. We limit the number of people we interact with, and still we're seeing very high rates of infections. So I think we still need to see sort of the data and the experience, but I think when it comes to the spread of the disease and thus sort of prevalence, it's much more than I would have sort of expected before, and seeing the severity of the disease, especially in the elderly and other vulnerable people, clearly, clearly there's an impact.
And here we have another good question by Dan about your vaccine program.
Yeah.
About, is there any news about the financing? I mean, there was some news, I think a month ago or something,
Yes, yeah.
regarding that you needed financing regarding the last clinical phases.
Yes.
Yeah.
So we're still actively having those discussions, but nothing has been finalized when it comes to new financing.
And let's take another one. That's from that's a hard name, Georgios. What will you say to people who say that we don't know what will the long-term effects of COVID vaccines be, so they wouldn't want to take it? So yeah, anti-vaxxers. Yeah.
Yeah. I mean, taking different. We don't know what the long-term effects of the COVID-19 disease will be.
Yeah.
It's a disease that keeps on evolving, keeps on surprising the scientific and medical community. We know that vaccines can be, and we know that some of these platforms have been, even though there hasn't been approved vaccines utilizing those platforms, many people have been vaccinated with them over the years, and we know that the safety profiles seem to be good. So yes, it's an open question. No one knows what it will look like in 30, 40 years from now, but we do have expectations what the long-term effects of COVID-19 will be, heart thing and such. So I would say that it's always a benefit, sort of risk-reward, but
yeah. And then what is left to be done before the phase 1 study can start? Is from Jens. I don't know. Yeah, how long your development is with your vaccine together with Advact. I think it's in that regard.
Yeah. So as Morten said, we hope to get it, or AdaptVac hopes to start the trial in the new year, and there's various things left to do, so just administrative as well. But yeah, hopefully we can start it at that time.
And then I will take a last question here from Kunal, who asks about how will Bavarian will meet or cope the demand for the doses of the vaccine needed. Yeah.
So one of the things that we have been active in is making sure that there would be production capacity. We've had those discussions with various companies if this vaccine is successful. As I spoke about and also Morten spoke about, it's a very effective or efficient manufacturing process. So we should be in a position, if the vaccine proves to be successful, to meet the demand.
Yeah, I think we'll say thank you for now, Tommy, and I look forward for the panel discussion later. And to everyone seeing with you, we're actually 198 viewers right now, so that's quite awesome. I mean, compared to like the normal physical event with 300. So I mean, we are really happy here at Synapse. But yeah, we'll take a small break, and then Thomas from the Danish agency, medicine agency, will come on. And yeah. Thank you for now.
Yeah, thanks everyone.
Yes. Welcome back, everyone, to this last talk with Thomas Senderovich, director at the Danish Medicines Agency, and he will take you through some of the regulatory processes that is required to introduce a new medicine to the market. So take it away, Thomas.
Thank you so much, and good afternoon. To this last talk, the only thing between me and you and gin and tonic, so I'll be brief. We're going to talk about the role of the regulatory agencies in approval of vaccines. We're going to focus in on vaccines because that's what we're talking about today. A little bit about our agencies to understand what we're working with when we don't work with COVID-19, which has been pretty much what we've been doing for the last 10 months or so. So we're around 500 employees, so we're sort of a mid-size agency compared to the size of Denmark. We're very big compared to other agencies in this field here. We authorize and inspect medicines, companies, and clinical trials in Denmark, the Danish market. We monitor the safety of adverse drug reactions, both from clinical trials but also from medicines and medical products on the market. As I said, we also authorize clinical trials. We also decide whether medicines for the primary care use should be reimbursed. So that's part of what we do as well. We also monitor medical devices that are available in Denmark, and we supervise adverse incidents involving medical devices. And then we also manage the entire pharmacy sector in Denmark. We appoint pharmacists and we inspect them and so forth. We have a few other tasks. So it's it's an all-in-one circus, you can say. So it's quite a lot of work. But we're going to focus in a little bit on what we do when it comes to vaccines. I think first I'm going to focus on the European Union here. It would take us too far to go through how they do things in South Korea, Japan, and US. So we'll focus on Europe. And basically, there are different ways by which you can get a medicine approved. You can either go through the centralized procedure, which is through the European Medicines Agency. I'll come back to that. Or you can do quite a lot of medicines are still approved nationally, either through a pure national authorization, a decentralized procedure, or through mutual recognition. These three to the right of the slide are the procedures by which the mostly generic medicines and non-innovative medicines are approved, whilst all really novel medicines, and including that in that field you have vaccines as well, genetically modified medicines to treat cancer, CNS diseases and so forth, they have to go through the centralized procedure. As a company, you can ask EMA if you want to go through the centralized procedure, but I can assure you these days, if you're not on the mandatory list, you won't get through because all resources are focused in on COVID-19 procedures on top of what we're doing already. So that's the overarching system we have. And so the way it works is you have the European Medicines Agency based in Amsterdam, which on behalf of the entire EU, plus EEA countries like Norway and Iceland, basically takes care of this process. But as you can see, the scientists performing these assessments are actually based in the national competent authorities, like the Danish Medicines Agency or the Swedish Läkemedelsverket, and others, the French here. And so we actually have the scientists performing these assessments on behalf of EMA. And all of these agencies, each of us, we have 2 members placed in the committee system under the European Medicines Agency. So the committee is looking at approval of new medicines. It's called CHMP, Committee of Human Medicinal Products. This is the central nervous system of the approval of novel medicines. In addition, we have the Pharmacovigilance Risk Assessment Committee, the PRAC, and they take care of all the safety follow-up of each medicine placed on the European market. So this is the procedure, and let's just have a look. Oh, well, we actually go one back, because once you have submitted all your data, and today it's all done electronically, then these committees, they go through the evaluation, and then they come with their recommendations, either thumbs up or thumbs down, or we're not so sure. If it's thumbs up, then the opinion of the CHMP is then sent to the European Commission, which then authorizes, submits, creates or sends out a marketing authorization, which then immediately grants the company an authorization to market the medicine across the entire Europe. They're not forced to market, and that's important to realize. They're giving the right to market. All right, so now we're going to focus in on vaccines. So we're going to look into fast-track vaccine development during a public health emergency. And this is what we have. And I completely concur with the previous speaker who said, this is not a flu. I remember when it all started, actually, we were on the forefront because I talked to my colleagues in the European Medicines Agency before the COVID-19 came to Denmark, and we discussed if COVID-19 could have an impact in the supply of medicines, because a lot of the active pharmaceutical ingredient actually comes from China, and if it then spread out to India and closed down infrastructure, then we would have a challenge. But now we have a pandemic, we have a public health crisis, and so we have to fast track this development. And as we are quite aware that we really haven't got a very good cure. We have the steroids that definitely have some impact. We have remdesivir, which is still conditionally approved, but is not a lifesaver. And that's it for the time being. There's something coming in the pipeline for the cure, but I think everyone is looking forward to having vaccines which can then ultimately help address this problem by introducing immunity in the populations and then gradually reopen the global society again. So during this fast-track development, what is very important, and as we've also already seen, is that we have a huge. I have to say, this is an epic format. This is unheard of. The amount of R&D, the amount of resources, the amount of scientific groups and companies looking at novel COVID-19 vaccines, I've never seen that before. And the speed has been, really phenomenal. And of course, that's exactly what also creates some concerns in the populations, because it can't go too fast. Normally it takes 10 years to develop a medicine and a vaccine, and here we are actually in about a year's time. This is unheard of. So in order to guide and foster that the best vaccines and those that can actually become real medicines, and I like to be a little bit of a particular, with, and when we have the panel discussion later on, my two fellow speakers can then beat me up. But I like to see clinical data, real clinical data. I like to see them, robust clinical data, before I say we have a medicine. Until that point in time, we have a promise. And it's good to have a promise. I'm not against promises. I just want to see them fulfilled to become real medicines. And that only happens when they're approved. And therefore, the early scientific advice that EMA has offered to regulators and the national agencies are really important to help guide them in this very difficult and undescribed landscape. Of course, there are plenty of guidelines, existing guidelines of how to develop vaccines, but science and technology develops faster than guidelines do, and hence scientific advice in which you, with the agencies, can discuss some of the scientific findings and the designs of the clinical trials are immensely important. So there are these opportunities right now. Then there's a special task force, a COVID-19 emergency task force, which has been put up, which can offer rapid scientific advice and help to design the best possible studies. And then you have this support for medicine and vaccine developers, and we also have to advise on how these developers can make sure that they have the appropriate quality of the vaccines. The quality is about how the vaccine is manufactured, that it can be kept under the right conditions, stability studies and so forth, and of course safety and efficacy. These are the drug development interactions with agencies. Now, under normal circumstances, in peacetime, if we consider COVID-19 wartime, then normally you start off in your pharmaceutical quality and non-clinical in vitro studies, and you have small-scale in vitro studies. You move into animals, and as we heard earlier, animal models are necessary. They're necessary for pharmacology and mechanism of action studies, they're necessary for safety studies, but they are not human studies. And unfortunately, I have to again be the party killer. Although we can celebrate good results in animal studies, I've been in this business long enough to see that only a small fraction of what turns out to be good in what is good in animals turns out to be good in humans. So only a small fraction of that. So that's really always the risk when you develop medicines. That's the attrition rate going from animal to clinical studies. And then you perform your clinical trials. I'll come back to those in a second. Phase 1 studies, phase 2 studies, and phase 3 studies. In this time here, under these circumstances, we have seen study designs compressing 1, 2, 3 almost into seamless clinical programs, which we, under normal circumstances, are not too happy about, because then it's more difficult to be very clear about the hypothesis that we want to test and so forth. But under these public health crisis conditions, we can accept having these compressed study designs. Then these data are submitted to the EMA, we go through the evaluation, and then you scale up. This is a very long process. This is 10 years. I mean, the evaluation for the European Medicines Agency under normal circumstances are 270 days. And that's because a lot of stuff is sent in. And very often there are questions back and forth, and only then do the manufacturers, the industry start the manufacturing, because it's very expensive. And quite frankly, you don't want to invest in huge manufacturing capacities if your medicine isn't approved. That's actually loss of investments, and no one really likes that. So that's under the normal circumstances. I'll come back to what happens under accelerated. Just let's focus a bit on the phase 1. Typically 20 to 100 healthy volunteers. When it's vaccines, and it's vaccines, not cancer vaccines, but in this case, vaccines against a virus, then everyone in these studies are healthy volunteers, or at least not ill with a certain disease. They can have comorbidities, of course, but they're healthy volunteers. And in the beginning, all you actually want to look at is, does it seem to work? And which you can do in phase 1 in a vaccine situation, but you can still look at immunity. Are there any serious side effects? Is it at all? Is it safe to go further into larger scale studies? That is what phase 1 can do. And when companies announce their results from phase 1 and say, oh, we have this fantastic result, then always remember these are 20 to 100, max 150 healthy volunteers, and there's a long way from a phase 1 result until you have a medicine. But, you know, every time you survive, every time, not the healthy volunteer, but every time a medicine survives to the next phase, we're always happy. We're, of course, happy that volunteers survive always. Then we're in phase 2, several hundred volunteers, and here again, you look at the common short-term side effects, because these are not long-term studies, and you look at the optimal dose regimen, because you really need to know which dose regimen, which dose you need to give. In vaccines, you basically ask yourself, is one shot enough, or do you have to give a couple of shots? That's the whole discussion. And the reason you need to give 2 shots with vaccines, of course, is that you need to have the immune system fully up and running, so that in many cases 1 shot isn't enough. Of course, it's easier. It's the target product profile, in particular if you're on the rural parts in developing countries, then it's really not always easy to get your subjects you have to vaccinate into the clinic or have to get out. So it is better if you can give 1 shot, but that might not always be enough. And then you come into the phase 3 and thousands of volunteers. And let me say it very clearly, what we're seeing these days here, these are, it's like the project of getting the first man to the moon. This is how big this is. It is the enormous efforts in getting all this off ground and landing safely and bringing them back, because we have 20, 30, 50, 60,000 volunteers in these studies. These are humongous studies. And of course, that's important to realize and to remember when we talk about, can we trust the data? Isn't this too speedy? When we have this amount of volunteers, we have a better chance of detecting rarer side effects, and we have a better chance of identifying efficacy faster. And that's the whole idea with these big studies. Now, of course, it plays to the benefit that COVID-19 hasn't been under control, and in many parts of the world, you've seen huge pandemic numbers. Hadn't we had that, it would have taken a long time to develop the vaccine. So, you know, even though we would all would have been, we would have wished to be without the pandemic, then of course having it out there in this size allow us to actually develop vaccines faster. So there's always some good in what otherwise is bad, but that's one of the reasons we can develop them with this speed. And then we also have to realize that a lot of resources, a lot of investments have gone into this, also from governments. And I think that's pretty fair. Under these circumstances, I think it's fair that we as country administrators and politicians say, we want the vaccines faster, we have to co-invest to a certain extent into this. So this is the very advanced digital transformed world, as you can see here, of what approval of medicines is all about. It's simply making the benefit risk assessment. It's pure and simple. Of course it isn't. It's not that black and white, but this is what it is about. It's to look at all the data, the totality of the data for the dossier for that particular medicine, and you take a look and say, are the benefits outweighing the risks? Is the efficacy we see big enough? Do we have side effects that are unacceptable? In the case of a vaccine, you most certainly can't accept to introduce diseases which are worse than the disease you want to cure, because otherwise you're actually treating otherwise healthy, or at least sort of healthy population. You really can't introduce something worse. It's of course a different discussion when you talk about a cancer. If you have patients with an incurable cancer, then of course you can accept significant side effects because the prospect for that particular patient population is very challenging. When we talk about vaccinating the entire population, you have to be pretty damn sure that you're not introducing significant problems. Of course, when you vaccinate, you can expect some reactions to the vaccine, local reactions, some pain, some swelling, some redness. You can expect transients, fever and so forth, but you shouldn't really have so much more than that. So that's the challenge. Then you also have to look at the mortality of the overall impact of the disease you're trying to prevent with the vaccine. So now to the accelerated development and approval. This is the view of the developers right now, or the regulators. This is a blurred, super high-speed train, and it has to stay on tracks and has to arrive safely at the station. So here, this is an overall. I have to say I stole these pictures. I didn't make them myself, so I don't want to take any credit for that. These are all stolen from the European Medicines Agency's homepage. So you can actually go in there and you can get much cleverer than listening to me. But I'll try anyway to explain the differences. You can see what I explained. To the left, the standard development, it takes forever, it takes 10 years. And to the right, everything has been compressed. And how can that happen? And what about the evaluation of this submission? So again, this is what I showed you before. You go in a fairly serial way. It's all in sequence: pharmaceutical quality, the in vitro studies, non-clinical research in animals, the phase 1, 2, 3 studies. You submit it, you evaluate it. Only once everything has been finalized and the dossier is submitted, you evaluate it, and then you scale up the production, and then you have some studies after authorization. You have your risk management plan, which is mandated under these circumstances. That's the standard game. Now we are in a non-standard situation. And what you can see, the big difference is a lot of this is done in parallel or semi-parallel. So you actually start with much more intense scientific evaluation and authorization earlier. And I'll come back to and this is possible under EU law. It's also possible in other areas, of course. And you also see that the large-scale production is started at risk before the authorization. So a lot of things are done at risk. At risk means at the risk for losing money. That's what it means. It's not at risk for the subjects in the studies. It is at risk for the company to lose their investments. It's also at risk for us as regulators, because we have to be lined up and invest significant resources in being ready and evaluating something which may fail. But again, we have decided unanimously across the board that of course we will do this because we have a global health crisis. However, there is no difference in the requirements for safety, quality, and efficacy there is no difference between the requirements in safety, quality, and efficacy between the standard conditions and the COVID-19 accelerated conditions. And this is very important for me to say. We are not shortcutting our scientific scrutiny during the accelerated review and approval times. We are not. We are lining up earlier, and we are conducting what is called rolling reviews. A standard review on top of this slide is you do all your development, you send it in, and then you look at it. During the COVID-19 crisis, you send in your data in batches during a rolling review, and you review it batch by batch, so you are actually getting it in live almost. This has to be agreed up front between the applicant and EMA, but this is what we do. And currently there are 3 vaccines undergoing rolling reviews, and this is public knowledge, so that's the Pfizer-BioNTech vaccine, it's the AstraZeneca vaccine, and it's the Moderna vaccines. And for sure the other ones that a little bit later in their phase 3 programs will add to this portfolio. And for each of those, you start looking at the quality package, the non-clinical data, because the phase 3 studies are just being finalized. And you have all seen the amazing results, which I have to remind everyone are press releases. So I wouldn't open all the Dom Perignon bottles right now. I would keep them in my fridge until later, maybe New Year's evening, because it's too early to celebrate based on press releases, unless, of course, you are shareholder, which I'm not. I'm working for an agency, so I don't get any benefits for these press releases. I'm sure some will do. So you can hear I'm a little bit critical to these press releases that a little bit prematurely start celebrating good news before we've seen the data. All right, anyway, so this rolling review will allow us to actually have a very shortened timeline. And actually, during these days, we have discussions between the Commission, EMA, and the heads of agencies. We are discussing how we can shorten the time from EMA opinion until we can grant a marketing authorization. The marketing authorizations that will be granted are conditional, which you can also go in and read more on EMA's homepage. This means under certain conditions, you can grant a marketing authorization earlier, even if the phase 3 studies are actually still ongoing. In a vaccine situation, they're ongoing in terms of follow-up, so they're following the study subjects. That means if we see something dodgy, if something happens, we can take the authorization back. And it's only valid a year. And then it has to be renewed. And at that time, the company has to submit more information. And then it can be renewed again for another year. It can then be made permanent if everything is fine. So this is a tool which will allow us to issue marketing authorizations earlier. I also want to say something else. What happens in US and UK with these vaccines are not approvals. These are emergency use authorizations. It's not the same. This is a real approval we're giving, albeit it's conditional. Whilst what you see in US under US law, and I guess it's the same with UK, which recently left the European Medicines Network collaboration because of Brexit, they have emergency clauses. I'm not saying that they're not deep into the details. I'm just saying it's not the same. Right, so that's it. This is what happens these days here, and this is the procedure that is ongoing. We have scientists from our agency deeply involved in these processes, and I'm happy to take any questions you might have.
Very good questions for you. But first of all, thank you for a very nice speech. It was some really great views to add to the whole vaccine development aspect. Emilia asked, how long are the studies you perform in order to evaluate the side effects from a vaccine? Do you conduct long-term follow-ups?
Well, let me start by saying I don't conduct these studies. These are the companies who conduct the studies. So I guess the question is to the companies. And so absolutely, the protocols, which, by the way, for these vaccines have been made public, which is also very good in the name of transparency, these studies continue for 2 years. So there will be, in the phase 3 studies, there is 2-year follow-up. However, as long as the vaccines are marketed, they are followed up by mandatory pharmacovigilance. So that means that healthcare professionals, they have to report adverse drug reactions following these vaccines. And we keep our eyes on this, and we have a national database in Copenhagen, and we have a European database, Eurovigilance, into which all these adverse or suspected adverse drug reactions end up, so we can track it as long as the medicines are on the market. But these studies here are 2 years. Now, if we see something during this period of time, we can mandate longer term follow-up. We can mandate companies to do something else. But right now, it's a 2-year follow-up for these vaccines.
Okay, and I think we'll take one other question. Also, I think this is really interesting, do you believe rolling reviews, so to speak, will become the new norm moving forward compared to standard reviews?
Nope. I don't believe that. Not unless we are willing to increase taxes in Europe so that we can hire more people, because this is a very resource-intense procedure. Now, when I say no like that, I want to add that what we're currently seeing and what I suspect will happen, not because of COVID-19, but because of the need for digital transformation, is that we will, in the European Union, we will become better at analyzing raw data. And so the more you can submit in a more seamless way and get analytical systems up and running, the less resource intense a lot of this will be. But right now, a rolling review is extraordinary heavy workload. It means we're basically pooling in expertise to look at these. But I have to remind everyone on that there are a lot of other diseases out there that need to be treated. So we can't just keep the rolling reviews as the focus unless significant resources would be added in, and I don't expect that that will happen. So the short answer is no.
Okay, great. Thank you. I think we will keep it at that for now, and then we'll take some of the other questions for the panel discussion. So thank you once again for your view. And for you at home, we will have a short break, and then we will be back with the very exciting panel discussion to come. So please, again, send in any suggestions you might have or anything else that you want to discuss in further detail. See you soon again.
have now reached the panel discussion, and we are thrilled to see that even more people are watching now. So, I would also say personally, this is the part of this whole where we can actually ask you about some of the really interesting thoughts we had. So we'll through it, and we'll take some questions, which has been asked the whole session. So we have a question from Neil to begin with, and that is fast tracking vaccines at the expense of safety tests and detection of side effects? And let's start with Thomas. Yeah, that is within your field, and then we'll elaborate a bit afterwards.
So the short answer is no.
Yeah.
Because we, as I explained, we have exactly the same criteria and demands for safety evaluation in the COVID-19 as we would have in a non-COVID-19 situation. I think the only thing to be very transparent about, of course, is that when these vaccine dossiers, when the data are submitted to regulatory review, we do not have 1 year or 2 years follow-up. If we wanted that, which could be a reasonable or at least a request, then we could all sit down and wait with the current status of closed societies for another 1 to 2 years. And that has a huge implication for public health and the economy as well. So when you make a decision saying we can accept, so the median follow-up for the last included would be 2 months.
Yeah.
But a significant proportion of these 20, 40, 60,000 subjects would be longer. Right, so we will have a very large safety database. But of course, we won't have 1 year's efficacy, we won't have 1 year's safety. That will come as we follow the vaccines subsequently. So that's the balance, quite frankly. And 100%, as you also know, I'm sure all the viewers will know, 100% sure of something doesn't exist in biology. So I think, again, this was a little bit longer, but that's the rationale, the thinking behind saying it's good enough. But there's no increased risk of adverse drug events or real safety problems popping up. And the last point I want to make, with one known exception that you may also know, the pandemics. But generally, you don't see side effects of vaccines popping up after 6 or 12 months. It's not the same as a daily accumulated dosing regimen where you see something. So that's also very important to remember.
Tommy, I kind of also wanted you, if you can add something, because you have more of the company perspective on it instead of just the regulatory. I don't know if you have anything to add on it.
Sure.
Yeah.
And just sort of to support what Thomas was saying, I think the regulatory process that I have put in place will be as rigorous and looking at safety and efficacy. The rolling submissions, I think, do a lot because you actually get the data real time and can sort of progress it rather than waiting for it all to be finished. So I think that will sort of cut through a lot of the wasted time, if you will. I would still, however, it is this sort of long-term data that I think we all will benefit sort of achieving, both in terms of durability of the response, but then also comparison of the different vaccines going forward. So I would hate that society here in the Western world or other parts of the world would be in a position where we just sort of accepted the first 3 or 5 vaccines and then that's it, because we need to make it possible to develop and commercialize other vaccines if they're proven to be even more efficacious, more durable, or cheaper to manufacture, or things like that. So I think that's, to me, that's the the next step, and how authorities and others will sort of view that sort of ongoing development of additional vaccines for this disease.
I think I will actually just take you a bit on that and ask you, Morten, because your collaborative vaccine is not in clinical trials yet and will probably come out later than most other vaccines. And personally, I would think that is kind of a downside, but it, of course, then you have to prove additional efficiency and stuff. But what is your own thoughts on coming a bit late in the game compared to the early vaccines, like the mRNA vaccines coming out. Yeah, hopefully in the beginning of the new year.
Yeah, the problem is, of course, that you need to measure yourself against or measure one vaccine against others.
Yeah.
And as there is a registered vaccine soon, hopefully, it will be very difficult to make a phase 3 because you have to give that vaccine as well. So it'd be very difficult until we have what we call a surrogate marker, where, for example, a threshold of antibody that you have to be above to be protected. Before that is realized, then it will be very difficult to start a new phase 3. I think I can add to the previous that the reason why it has been able to go so fast as it has is that all the public money that has been entered into vaccine development has de-risked the development for the commercial companies. And that's why it's going so fast. They can start scale up while they're doing phase 1, and they can do filling while they're doing phase 3. So that's why it's very rapid at the moment.
Yeah, I mean, it's really nice that things are going so fast as it is right now. I mean, hopefully everyone wants a vaccine right now. But yeah, I think we should take another question. And that is from Eugene, who says, given that current candidates announced by Pfizer and Moderna requires ultra freezing, how can we secure distribution to remote places that have limited facilities? Was it you, Tommy, who, I mean, it was in your talk, so I mean, maybe you could elaborate a little bit on it if possible.
Yeah, sure. So first of all, there is a Merck vaccine against Ebola that also requires, I think it's -60, -70 degree storage, and it's been successfully deployed in the Congo, which is a fairly rural area and then very difficult to get to. So it can be done. Having said that, those vaccinations are, of course, on a very different scale than we would be looking for COVID-19 vaccinations. I think both Pfizer and Moderna are very good companies. There's been a lot of development. I know specifically for Pfizer, because it's public, how they've been thinking about transport and storage. So these are all engineering things, and engineering you can always solve by throwing enough money at it and resource. So I think it will work out. Having said that, I'm sure there will be other vaccines coming which will be hopefully easier and less expensive to construct the supply chain for.
Yeah, I actually can't remember. I mean, I think you mentioned it, but where is your collaborative vaccine? What temperature is that coming? Like secure, biological. What is it called? Yeah. Stable at?
Yeah, at the moment we have it at -20, but we are investigating at both in the fridge and also in ambient temperature, and it actually looks quite good. But I also know that Pfizer, BioNTech, they're looking at freeze-drying their vaccine, which would then mean that they can store the vaccine at higher temperatures.
Okay. Yeah, I mean, it's quite interesting. And also, I mean, you mentioned Moderna. I mean, so as it is for now, I mean, I understand that Denmark does not have an agreement with Moderna, right?
Well, no. I mean, the way it works is that the European Union signs on behalf, and then there are 5 days to opt out.
Yeah.
So we just have a system in Denmark which it takes just to make sure that the politicians get their time. So I mean, I can't speak on behalf whether they would opt out, but that would be the first opt-out they've done so far.
Yeah.
So I would be surprised if they do so.
Because, I mean, I think I read the other day, I just looked through your webpage and all the agreements with, Johnson & Johnson and Pfizer and
AstraZeneca.
Yeah, exactly. And maybe I've just, didn't see.
No, you haven't overlooked it. We have those and Sanofi, GSK, and CureVac. Moderna was signed today with the European Commission, and then there's a 5 days opt-out.
Okay.
And within those 5 days, the Danish government will inform us in my agency. Yes, we will not opt out, or we will, but I would be surprised. But let's see. And in that case, we're in that game as well.
Okay.
So just to add, and because of this, we have actually a menu once they get approved and arrives, we have a menu of vaccines with different technology. And even for those that are currently have stability data on -70, because it's accelerated, you can't rule out that stability data would arrive later on, which would show that you don't need.
Yeah.
But because everyone is waiting, everyone in EU is currently planning for the ultra cold chain of -70 for messenger RNA vaccines. We all hope that it won't be necessary. And to answer your question, it may not be. It's the messenger RNA vaccines that first will be coming out to other areas of the world where logistics are more difficult to handle. It's not just whether you can have a freezer. It has to be kept like that all the way out to the end. And at least for the Pfizer-BioNTech, you really can't have it out of -70 for that long. And in particular, if you are above ambient, which you will be in some countries in Africa, you run into problems.
Yeah.
So there will be other vaccines. And now I have to say something which diplomatically isn't very very good. But at least from my point of view, I haven't seen the clinical data from and long-term follow-up from the Chinese or the Russian vaccines. So I really hope that both the Chinese developers and authorities and the Russian vaccine developers and authorities put the same scrutiny in, because they might very well be supplying parts of the world that currently won't be supplied with these vaccines.
Yeah. Yeah. But yeah, let's take another one. David, do you have one? So yeah, that's from Anita. How will you make sure that a vaccine developed just in 1 to 2 years in contractation to 10 to 15 years will be safe enough with minimal side effects? Let's start with.
I think we've answered.
Yeah,
I
also feel. We have
just to repeat it, but these questions keep popping up because they're important questions. So let me remind ourselves that the underlying number of subjects in the pandemics which had this problem, as a given example, there were 6,000 in the total database for pandemics. Similar vaccines, other vaccines, we're talking about 2,000 or 3,000. In these studies, per vaccine, we have 20, 40, 60,000. These are huge studies.
Yeah.
So that's one reason why we feel rather confident.
Yeah.
The other reason is that we apply exactly the same scrutiny, review scrutiny. And actually, the reason we can review the vaccines on a shorter time frame is that we start earlier and we review these data as they come in, as I explained, in rolling reviews. So that's the reason, and we put really a lot of people on this. And then I want to say, in Europe, we have a rapporteur, which is one country, which does the review on behalf of EU, and a co-rapporteur, 2 independent teams review, and then we have peer review. It's an extraordinary scrutinizing process. That's why I feel we are absolutely as confident as for any other medicine we approve. And so the development shortage that you've seen is also, as you pointed out, the huge amount of investments that are gone into this. And that everyone has been focusing on the same ball on the field, on the playing field. So I think there are many good reasons. We can't do this every time we want to develop a new medicine, because then we'll run out of resources. But in a public health crisis where everyone is on the same challenge, then it's possible.
Yeah, and maybe just to add to that, because I think Thomas made an important point in terms of the funding and how that makes it possible to circumvent some of these steps. Because those phases, actually, it's also for the pharmaceutical company. They're not going to invest into manufacturing a vaccine before they, it sort of passes and there's market approval, because you don't want a stock of vaccine that you can't sell. You don't want to initiate a phase 3 trial before you have sort of the phase 1 trial almost done, or even phase 2 trials. And here, things just moved very quickly because you could take the financial risk of investing. I think Pfizer invested $2 billion in their vaccine development. They did that, and then some of the other companies got very significant funding mainly from the U.S. government. So I think that there's a lot. It's not that that 10 to 15 years all do because you're just following up these patients for such a long time. There's also a time there where you're not making decisions because you don't want to take the financial risk.
Yeah. Yeah, I mean it is. Yeah, yeah, it's totally understanding. I mean, there's so much money in this right now. Also, I mean, the U.S. government had also invested, yeah, quite hugely, right?
But so did the EU
Yeah,
as well. So the advanced purchase agreement that we're doing
yeah,
is de facto a de-risking upfront payment.
yeah.
So it's also de-risking.
Okay.
So U.S. has been a very generous beneficiary, the EU as well, and other countries.
Yeah.
So that's the whole point. And of course, you can't do this all the time
No.
because and basically it's a public health crisis. You can allow that to do so. But I think the on top of that, the fact that we get the vaccines, what we also talked about during the break is that you also now have a platform. You now actually have a scientific vaccine platform which has been created so that for future challenges, we can expect that we will benefit from the investments that we've seen here. And that's an add-on benefit for everyone.
Yeah, I mean, it must be amazing for a person as you, Morten, like seeing so many scientific things like who has been talked about for years now actually going out from academia to being part of startups or like being used for vaccines. I mean, yeah.
It's very, very exciting
Yeah.
to be in the middle of this, because all the technologies is being put into the pot, you can say, and stirred, and now we see how the flavors are coming on
Yeah.
later on, and that's very exciting. Yes.
But yeah, is there a good question? So, how do you see vaccine of the world population develop when a vaccine is ready, and what action do you think are necessary to eradicate COVID? So, I'm just
Yeah, what we don't know at
Yeah.
the moment is whether the vaccines that are currently in the press, whether they will reduce transmission as well. They have an endpoint which is the symptoms at the moment. Actually, we don't know whether it's also very effective against severe COVID. We have some indications that it probably is, but what about transmission? Can it also hinder transmission of the virus? That's going to be very interesting to follow in the next half year.
And that's also why, just to be absolutely clear, the general behavior that we have put in our society must not change
Yeah.
for a long time until we see high-level data or meager data that you actually are changing the nature of the pandemic. It would be wrong to think that because I got a shot, I'm not transmitting. Exactly as you say. I have a certain percentage of being protected. It will not be 100%, but I could still transmit and so forth. And that's something we will learn as we go on. So it's an extremely important point. And that's why the good question here, no one can answer. We can speculate.
Yeah.
But what it will take is an immense effort. And let's be clear, very few viruses have been eradicated. You can keep it under control, but eradication, it is not as easy. That requires global flock immunity, and that will not happen in 1 year. It won't happen in 2 years. It will require more than that. That's my guess before you have a global control of it.
It is. I mean, the only virus that has been so far eradicated is smallpox.
Yeah.
I mean, it's only in freezers. But that is because the only known natural reservoir of smallpox is humans. There is no animal reservoirs. And not going into various other reservoirs on this, because it's been talked about so much. But I think that's another thing that we need to understand, because if there are, apart from humans, other reservoirs of COVID-19, it's going to be very difficult to eradicate.
So maybe the goal isn't eradication. Maybe the goal is meaningful control, which is also good enough. I mean, we can live with other viral diseases on a good control where you reduce mortality and morbidity, but it's not eradicated. So I think that's probably a more reasonable. It's a good question, but it's a question we can easily better answer.
Yeah, I mean, it's hard to say when. I mean, when the world will be totally free of COVID, or if it probably never will. But I mean, at least our living circumstances will probably be less, less affected. Is there a last good one? Okay, so Will you all be getting the first upcoming approved vaccine when given the opportunity? I mean, who should we start with? I mean, let's start with you, Morten. I mean, you probably would, yeah.
The thing is that it will not be given to me first instance because I'm not in the target population, but I would say yes, of course. And the thing is, and I think I can say what you mean, and that is normally we think about vaccines as something that saves lives.
Yeah.
Actually, in this instance, a general vaccine would create life on the globe again because we are all in lockdown and have no life, you can say. So a vaccine will create life again on the globe.
And what about you two? Would you take it as well?
Yes.
Yeah.
Absolutely.
Yeah.
Not a second hesitation. But again,
I mean, you have also approved it, right?
I have also said I'll take the first shot, but I probably won't be first in line, as you say.
Yeah.
So unless I sneak in front of Søren Brostrøm.
Yes.
Yeah, but I mean, if there's is there any like really, because otherwise I think we are on time, and then I will say thank you to all of you. I mean, I really enjoyed it. I felt we put together like an A team about COVID and fast tracking vaccine development. I mean, I have at least really enjoyed all your speeches, and we have in these COVID times. I mean, I would like to give you my hand and say thank you, but I mean, there is a small present behind your chair. That a bottle of wine. I mean, maybe instead of your gin and tonic later, you can enjoy a glass of wine. At least you can think about it. But I mean, yeah. And then I will talk to this camera and yes, and say thank you to all the viewers we have had today. I mean, we really enjoyed it. And I think at the highest point we were actually about around 240 viewers, which I feel personally is amazing. I mean, especially in these digital times where we cannot hold a physical event, I feel, I mean, this attendance is amazing to see, and all you life science students being so grateful for this opportunity. And then I will show you a bit about what Synapse has planned in the next couple of weeks. Firstly, we have a first job in life science, where there will be some speeches about both from speakers, both from people who have recently taken a job and how you actually get a job. And that will be on the 10th of December. So that is going to be an interesting one. And then we are our hubs in both Lund and Aalborg has also something interesting upcoming. Firstly, Lund has has an event about consulting in life science coming up, and where BCG and will be present, and there will also be talk about the regulatory affairs and the clinical development. And lastly, our hub in Aalborg is having an event about sustainability in life science, where there will come different speakers within sustainability and talk about how they ended up there and what they are doing within this field and making the world more green. And then I will say, yeah, thank you to all of you. Again, it has been amazing and having all you being here. So yeah, and with this, I would end this end the stream and say goodbye. See you next time. Yeah.